Okay, then we can start up. Well, welcome everybody in the on this world's next meeting on the bioinformatics meta analysis on the ESKAPE pathogens. And today we will focus on the standard operating procedures as well as the use of the software. And I think, yeah, Krishna, it's up to you to further explain it, to take the lead in this meeting. Maybe Jay, you want to add a few introductory words?
Dear colleagues, thank you very much for joining the meeting. But next time we will organize in such a way that we will come very fast. The SOP was prepared by Krishna. So we have two parts of the SOP. The first part is being today we will discuss about that. He will give us a PowerPoint presentation and tell us how to do the things. And of course, the SOP, the complete SOP, we will take 2 or 3, 1 or 2 days to complete it completely and send it. Of course, we'll share the PowerPoint and other things. And thanks for answering the choice of the microorganisms. Maybe we can also refine that today or in a near future. Okay, with these words, I hand over to Krishna for the demonstration and to show us what we need to do through the PowerPoint. Yes, Krishna.
Thank you, Martin, and thank you, Jay. And good morning to everyone. Good afternoon, good evening. So, because people are joining from different parts of the world. So, with this brief introduction given by Martin and Jay, so let us start today's discussion on bioinformatics tools that needs to be used in our global study on antibiotic resistance in the ESKAPE pathogens. So what I'm going to do is I'm going to share my slides. I hope the slides are visible.
Yes.
Yes. So in this particular meeting, we will be discussing on the SOP, the standard operating protocols to conduct this analysis using different bioinformatics tools. We'll also be demonstrating different tools which are necessary to mine antibiotic resistant determinant from the ESKAPE pathogens. So this team will be coordinated by Martin, Jay, and myself. And so we discussed many things in our last meetings, why this particular study is important, what is the difference that is going to make, and how those outcomes of this study will help us to contain the spread of antibiotic determinants in different environmental settings, including human beings, animals, and the environment. So, and how our study will also helpful for the policy making. So what with this particular the brief the question in mind, we have asked many the sub questions, and to answer those sub questions, we have prepared a standard operating protocols, and we have named it as Handbook of Genome Analysis Protocols for the Global Study of Antibiotic Resistant Determinants in the ESKAPE pathogens. So this is prepared by myself and all our coordinating teams, which includes Martin, Jay, and Pratisakar. So this particular the handbook contains what it contains. So I will be sharing these the handbook with you, so that it will be a ready reckoner for all of our AMR ambassadors to work on the assigned bacteria to mine different antibiotic resistant determinants and put it in appropriate manner, which is presentable and publishable, so that we can make good study out of that. So this particular the handbook contains the preamble for the study. What is our objectives? The preamble and the objectives of the study we have already discussed in our previous meeting. And mainly the guidelines is what are the guidelines we need to follow to obtain the genomic sequences and the metadata from global repositories, and how we can retrieve these informations which will be useful for our analysis, which includes both genome data and the associated metadata, which contains what is the name of the isolate, from where it got isolated, and when it got isolated, and what is the sub information that has all those metadata. We will be guidelines are available in this particular handbook, and once the data has downloaded to our local machine, the next step is to analyze this data for antibiotic determinants. So as I mentioned in the previous meeting, we will be using the open source the tools which are available in the Use Galaxy portal. So this particular handbook contains the guidelines for the bioinformatics analysis and how to import the downloaded data to the Galaxy, and how those different tools will be executing, what are the different tools which are available, and how those tools will be executed to do the in silico analysis of these antibiotic determinants in these pathogens. And once the analysis has been completed in the Galaxy tool, how to retrieve the analysis reports. Once the analysis report has been retrieved, next we need to focus on our downstream analysis, where we will be doing it our different kinds of statistical tools, algorithms, and report presentation, graphics preparation, the charts. Those things are described in the downstream data analysis. And finally, this particular handbook also contains two other sections which are helping our ambassadors to get to know about the analysis in detail. So we have a frequently asked questions and references to the tools and different algorithms that we will be using it to mine antibiotic resistant determinants in the ESKAPE pathogens. So the next is the demonstration, and in the demonstration part, we will be specifically focusing on the global database that I'm discussing, that is the NCBA pathogen detection database, and how to log into the database, what are the different ways by which you can retrieve the data, and once the data is retrieved, how to clean the data to get the good information out of that. And those data we will be importing to the Galaxy tool, and in Galaxy tool we are going to select different tools which are helping to mine antibiotic determinants. We will be knowing how to run or how to execute these tools, and the execution is done, how to retrieve the result. So this is the part we are going to cover it in today's demonstration. With this small introductory remarks on the handbooks, I'm requesting Prithvi Sagar to demonstrate the different tools. Prithvi?
Yes, sir. Am I audible?
Yes, you are audible.
Okay.
What I will do is I will stop sharing.
Yeah.
Yes.
My screen is visible now.
Yes, go ahead,
Yes.
please.
Yes.
Yeah.
Yes. It's
Okay. So first part of today's demonstration is about the retrieval of the data, both the genome data and the metadata from NCBI. So in our SOP that we are going to share in shortly. So here it has given all the details. So today we will go step by step. Now first thing is the retrieval of the whole genome sequences of ESKAPE pathogens. So that will be from the NCBI pathogens. The link has been given in our SOP. So once you enter the provided link, you'll get this page. Here you have a search engine. So here you have to type the the strain bacteria of your interest. Now I'll give Enterobacter species and give enter. So here once you type your bacteria name and give enter, that will lead, or the SLED browser will open all the available genome data in the database related to the organism of interest, and then you have to choose the column. So here once your page opens, so here this is the Enterobacter, this is for Enterobacter species. So all the data for Enterobacter will be coming. First thing what you have to do is here you have to select the columns for metadata. So down here you have the metadata. So here there is an option called choose columns. So first you have to go to this choose columns. Then here you have to select the information that is required for you. So here we have provided the data which and all we have to include. So it includes strain, isolate, location, assembly, collection date, isolation type, host, AMR genotype and EST phenotype. So here you can select it. So if some other data is coming, you can just drag it and you can remove it, or again you can drag it to inside. This is how you can select the columns. Once you select the required columns, just give OK. So then you have to go for download. So here there is an option for download for both metadata and assembly. First you click on download. Then here you have two options. In the data type you have table and assemblies. So if you click on table, then the type of the table what we require is in Excel format. So give table and Excel in CSV format. Here you can see that there are 8,359 isolate records available for metadata. You have to just download it. So when you click on download, it will take some time and it will show the location, the file, wherever you want to download it. You can give save there. Then again, to download the assemblies, go for download, click on the assemblies. So now here it will calculate what is the estimated size of data available and also the available records. Now we can see that there are only 4,574 records of genomic data is available, whereas your metadata had 8,000 odd records. Again, for the assemblies, click on the download button. So it will take, because the size is large, it will take a lot of time to download. Once you download both data, then we have to go for cleaning. So next step is data cleaning and preparation. First, you have to extract the genome assemblies from the archived file. So I have already downloaded it. So once your download is complete, you will get a file like this. Its name is genome assemblies genome pasta. So here you just have to extract the file. Once you extract the files, inside that folder you will have the individual genomic assemblies that will be coming. So this data we will be using for analysis in Galaxy server. Also the metadata.
Priti, one
Yes, sir.
question.
Yes, sir.
If you open the folder, there will be that much entries. For example, there are
Yes, so
hundreds,
actually,
hundreds.
yes. So here the file, the whole data, all the genomes will be downloaded in single archived file. From
Okay.
there, we have to extract individual files. So we just have to extract that.
No, that
Again,
I
we need.
understand. But for example, if there are now I see only 5
Yes, sir.
files.
This is because it will
For
take a lot
explanation.
of time to
Yeah.
download the whole data. So
That
it's
means you will get thousands of data,
yes, sir.
thousands of files.
Yes.
Okay.
So
That's what we
that's
want to
what.
clarify that.
Yes, sir.
When you
So
get
here,
thousands of files,
yes,
we
for
need to
this
have that
particular.
much.
Yes, sir. So for this particular enter vector only, we have about 4,574
Yes,
genome assemblies.
correct.
While
So
for
what I want to tell to others is that there are 4,500 files will be in that folder. So you need to have that much space in the computer,
yes, sir.
right?
Yes, and also uninterrupted internet is required, because sometimes even in middle of the internet goes somewhere,
Mm.
then the whole downloading process sometimes it gets interrupted. So again, we have to keep for running. So there is a uh, problem with that also.
Mm. Thanks for the clarification.
Yeah.
Please
Yes,
proceed.
sir. Thank you, sir. So once you extract your data, then next step is to clear the metadata. So here, when you open the Excel file which is downloaded, so here so here we can see the columns, whatever the columns we have selected. So only those information will be downloaded. So here our focus is on assembly. So when you select the assembly, you can see that for few strains there is a assembly number, whereas for other strains there is no data. So that means these are the records which are having only the metadata, but they don't have the assembly. That means the genome data is not available in the database. So these isolates are not important for us. We will be focusing only on the strains which are having both metadata as well as the assembly data. So now to clear these isolates which are not having any assembly data, we have to clean them. So just you have to click on the assembly column, then go to filter. and then filter, you have to deselect all. And when you go to the bottom, you can see there's an option called blanks. So click on the blanks and give OK. So now you will get only the isolates data which are having only metadata but no assemblies. So these isolates we have from our study. So just you have to select them. So I suggest don't give control all and delete. It is better you drag them and delete the rows individually, because yeah, so much of data. Just click on right click, give the option delete the rows. Now again you go to data, and if you deselect the filter, you'll get only the isolates which are having the metadata and assembly details. So if you go down, okay, here there is 4075 minus 1, the headers, you can say it is 4574. So it is same as what we saw here when you go to genome assemblies. So the number of records that is available is 4574. So this number and our metadata should match. That means our data is now clean for our downstream analysis. Just you have to save it and close. So this is a whole part with the data cleaning. And then next step is the use of Galaxy. So
Prithvi,
yes, sir.
you can wait little bit, and if any of our ambassador has any clarification or doubt with the data extraction, the data cleaning,
Okay, sir.
they can clarify here itself.
Yes. So anyone has doubt, they can please ask, or they can also give it in comments in conversation. We can.
No questions.
Yes. Go ahead, Prithvi.
Okay. So next step is for the Galaxy. So the Use Galaxy has different links. You can accounts like usegalaxy.org, or you can use usegalaxy.org.au or usegalaxy.eu. So I'm the separate accounts for each pathogens, and we'll be sharing the credentials, the username and password for each pathogens to the groups that will be assigned for their analysis. So for Use Galaxy, you just have to give your credentials. So this is the Use Galaxy team, the page. So the right hand side, you can see the history. So this is the place where all the details of your data and analysis will be shown. And to the left hand side, you can see the tools. So there are various tools that are available for from assembly to annotation, mapping, all downstream analysis. There are multiple tools. If you can click one purpose analysis, you can see multiple softwares that have been provided. And first thing is how to upload. So here on the laptop you can see the option called upload the data. Click on that. So usually for one or two genomes when we work, we'll go for regular upload. Now because we are dealing with the large data set, we have to go for an option called collection. So click on collection, and then now my files are in local, so go for option choose local files. Then you have to open the extracted files, select all and give open and give start. So it will take now, because only few genomes are there, it will take only a few seconds to few minutes. But since we will be dealing with thousands of genomes, it will take a lot of time, and a proper good internet connection is also a must for uploading the data. So now all the genomes are uploaded. Then we have to give the option called build. Click on build. Now here you have to select all the data. Once you select it, you can give a name for your collection. So here I'll give just AMR insights. demo, and you create collection. On the right hand side, you can see now your collection. If you click on that, you can see all the genome data that has been selected for this collection. So this is your AMR inserts demo is a collection of all the assemblies that you have been selected. Now we'll start with the analysis. Okay, first software we'll be using and that we are demonstrating today is called Abricate. So we just here in the search tool, you just type Abricate. So you enter. Here the tools with in Abricate there are Abricate list, Abricate summary, Abricate and file utilities. So we have to use Abricate. So click on Abricate, and here you have single data set, multiple data set, and collection. We have the collection. So click on collection. So when you click on collection, since now we have only one data set, automatically that data set will come, and then you have to go for advanced options. Here you have database to use. Now we are using three different databases. One is card, then we have plasmid finder, and another one is VFDB. So first we'll go for card. So click on card, then rest all the parameters we will keep default. And if you want an email notification, you can select this option, or else just simply you give execute. So it will take some time to run because we have taken only 5 isolates genome data, so it will run in a few minutes. Meanwhile, I'll show for another one. Two more analysis, that is with the same option you have to go for dataset collection, select your collection, then go for database to use. Here you have to give plasmid finder and VFDB. So once you click this, with all default you have to just give execute, then the analysis will be run for the given command. So I think it's taking time.
For our all AMR ambassador, I would like to inform that the database that we have selected in this, the card that is called as comprehensive antibiotic resistant database, using which we will be maining the antibiotic determinants in the ESKAPE pathogens. So that will answer the the key question that we have. What is the prevalence of antibiotic determinants which are there in the ESKAPE pathogens? We will be using the CAR database to exploit that. The second database we will be using is the plasmid finder. By using the plasmid finder, we will be getting information on what are the mobile genetic element it has. The plasmid is one such mobile genetic element. It will carry antibiotic determinants. And the third one we will be using is the VFDB database, that is called as virulence factor database, through which we will be mining different virulence genes which are there in this particular pathogens. So these three databases are really helpful to answer different research questions that we have asked. Yes, Prithvi.
Yes, sir. So now I think the analysis for this here you can see the data number 7. 7 is that we have run for card database. Then 13 is for plasmid finder, and 19th one, this is for VFDB. Now you get the data. If you click on this, you will get the results for individual files. Now we have to compile all the data into a single file. So for that, what you have to do is there is an option called Abricate summary. So this will combine the aggregate results into a simple matrix of gene presence absence. So this is the data that we are looking for. So click on this. Once you click on, go for the dataset collections. So first we will go for card analysis. So click on the 7th one. You can always refer these numbers, and you can note that which number is has run for which analysis. So keep track on those numbers. That will help you in your downstream analysis. So 7th one is for card database. So just click on that and give execute. Same way we can give for plasmid finder analysis and also for VFDB. So again, this will take some time. So this is only for 5 genomes we are analyzing. So when we go for our actual analysis, I think it will be taking, sometimes it might take overnight time. It depends on the server load and the genome size, the data size that we are going to analyze. So the analysis has completed.
So, sorry, Pratik. One thing you can also explain. For example, if they log in and load the genome data, if it
Yes, sir.
takes overnight, we have a
Yeah.
provision to close it, and it can automatically do the rest of the analysis.
Yes, sir. When once you give the job command, then you can
Then
log out and go. It will perform its analysis.
automatically it will do.
Yes, yes,
That's
it will
more
do.
important. That's more
For
important.
upload, you can't log out.
Stop.
Only for
Yeah.
analysis we can. But when
Yeah.
you're keeping the upload, the server should be in the login stage only.
Correct.
Only for analysis, yes, we can give command and we can log out. Then the server will do its job. It can be done.
Correct.
Yes.
Yeah.
So
Correct.
here for the analysis now summary. So 25th one is for card database. So just click on that, and here you have option called download. Just give the download. Now to your location, this will be downloaded in tabular file. Give save. Just go to that. Now here the data will be downloaded in notepad file. So you can, what you can do is you can uh, create a Excel file for name card analysis. Just open the Excel sheet and also the data. So just here click select all, copy, come here and paste. So this is for card database. You can see for the genomes that we have selected, it will give the numbers that targets that have been identified in each bacterial genome. It will first, you can see the name of the genes, and it will show whether they are present or absent. So 100 means it is completely present, and if the score is 97, maybe there are some nucleotide variations. Or if it gives the dot, that means in this particular strain that gene is absent. The same way the data can be downloaded for VFDB and plasmid finder. So this is the first stage of task that we have to finish now. Yeah, if anyone.
Sorry, I have a question.
Yes.
How do we remember the numbers like 7, 25? So because there are different jobs going on, one for plasmid finder, the other for card. So
Yes.
Can we see that which one is, like, if you forget what is 27, what is 5, what is 28?
Yes.
Or 13? Yes.
Yeah, see, when you first give a execution only, you can see that, see, now this is the for the last execution what I gave. So it will give the input what it is, 19th one, that is the applicate on collection 6. So that means you can take a screenshot of this summary page, and you can save it in maybe one you can maintain a word file or a PowerPoint, where you can see that each that run, you can see the numbers. Other than that, I think there is
There
no
is,
way. Krishna sir.
Prithvi,
Yes,
there is
sir.
one more option called as the information. If you click on the information, that will provide you what is the different tools you have run. Yes.
Okay. Yeah.
Okay, thank you.
Okay. Okay. Yeah. View details. If you click on view details, it will give the dataset information, whatever that you have done.
Yes, what you have run, and those informations will be available on that. But we suggest you to write down what we have run. So it is easy for us to have a kind of a documentation in our hand. Maybe so
Thank
that
you.
will, yes, yeah.
Yeah.
Thank you, Mash.
One point I want to make here is that, see, when you have run here, the number has started from 5. I mean 6. and then it has went to 7, then directly 13, because that numbers are, when you click on this, these numbers are there, so these will be counted. So you may not get the continuous number when you're handling the large data set collections. So it is better to write it down the numbers, because it is not in a continuous order. So they will go from 13 directly to 19, then again 25. So in between the numbers are there. That is the data number that will be given to the individual files, data files.
Okay, okay, got it.
Yes. Any more doubts?
Can you go to the Excel sheet once? Excel sheet with a card.
Yes, sir.
We would like to inform our ambassador that some of the antibiotic determinants are there in a multiple copies. So if such genes are there in a multiple copies, it will appear twice. So maybe you can look at the one example somewhere. I saw it. Can it?
Yes, sir.
Yes.
Here, now I have
Yes.
selected. You can
Yes.
see that it is
Yes.
given 1, 2, 3, 4, and 5 times. That means it is.
So the copy number is 5 times. So it's a 5 copies of ERMRA gene that exists in this particular organism. And also when we do the executing the tools, we have given some parameters. I hope everyone has observed that the parameters, we have kept the default parameters, that is 80% of the identity with the 80% of the genome coverage. So that is the default. So if any bacteria is meeting that criteria, and such genes will be appeared in this particular the Excel sheet after the analysis. So if anything below 80%, that is not that significant. So maybe that, so it's better to avoid that. So here you can see here, minimum identity 80% with a DNA coverage of 80%. So this is the usual cutoff and the threshold value. So most of our analysts will use, even us. So any values with the more than 80, we can put it as a plus sign or present sign.
Okay, sorry, I have a question. Please, can you go to the Excel sheets?
Yeah.
Why are we having just 5 results? Is it that we have just done the first 5 genomes?
No. Actually, we have uploaded only 5 genomes today, because for the uploading of the data,
Okay.
it will take a lot of time. So this is, since it is a
Okay.
demonstration, we have uploaded only 5. You can see here the first collection. So it has only 5 data sets. That means
5
5
genomes.
genomes
Okay.
are there. So when you
Okay.
analyze for 100, you will get the result for 100 genomes also here. Whatever the number you're taking, everything will come together in the collection.
Okay. All right. Thank you.
Yes.
And if you have any suggestions, any clarifications with respect to the tools and with respect to the kind of data that we need to select, with respect to uploading, with respect to data cleaning, to maximum extent, we will help you with respect to that with the knowledge that we have. So what we will do is we will work in a group, in a coordinated, in a cooperative fashion to get maximum result with this data, with a publishable one.
But uploading that data, if anyone is facing any problems, I think they can let us know. We can find some alternative ways to upload the data. So maybe the coming days we will update with any further developments that we do.
Prith, I have a question that how much time will take, for example, if we have like 4,000 genome together
Yes.
for Applicate, maybe for uploading only. I know that the downstream processing will be continued without, even if we log out, but I'm talking about when we upload the genomes, how much time it will take for like 1,000 genome.
The time to upload the data, it will definitely depend on the internet speed, how much it is there, and I think it will take a lot of time.
I will answer this. So with our work that we are carrying out, and approximately with around the speed that is called as we are getting around 70 to 100 Mbps per second speed. With this particular field, we could upload around 1,000 genomes within 2 hours time.
Oh, this is good. This is really
So,
good.
yeah, and again, because we have lot of data fluctuations in here, we could do that. So there is another option that Prithvi was mentioning. Right now, we are uploading it from the local machine to which we have downloaded the data. There is also an alternative option through which that is called as file transfer protocol, FTP protocol, through which you can upload maximum amount of data with a limited time, with a limited internet capacity. So for that, there are several tools. One is called as FileZilla. I think many people have heard about the FileZilla. Another tool called as Putty. So these are the two tools which will help you to upload the data from your machine to the Galaxy tool with a limited time. Maybe another,
Okay.
if you're facing a problem with uploading this 4,000 data, what you can do is you can split the data into 2 or 3 parts. Each one will be having around 1,000, so that you will be having around 3 to 4 files. Each files you can upload it individually, do the analysis, get the result done, delete the data sets already there in the use Galaxy, upload the fresh one. So that can be done. And the use Galaxy with this particular license version, we are getting around 200 GB spaces. So, but there is an option to request this use Galaxy data provider to upgrade the data storage. If that demands, we can do that also.
I see. Thank you. Yeah.
Thank you.
So for I think only the Enterobacter species has 4,000 data sets. I think Staphylococcus it has 30,000 genome assemblies. So I think as far as I know, that is the huge data for Staphylococcus. It is the one having the maximum assemblies available. So almost 30,000, 175 odd assemblies are there, and then that will be followed by other bacteria. Least one is Enterobacter species, that is 4,000 odd assemblies.
So we request you to select the organism based on your preference, based on your prior experience with the pathogens, as well as the infrastructure capacities that you have, your system capacity, your internet capacity, and ability to handle multiple sequences at a time. So any specific questions, any clarifications, any input, any suggestions, we can discuss and we can have a productive work together.
And once again, this is the part 1 of the work. We have part 2, but we'll do it after finishing this one. So without any confusion.
Yes, the part 2 will be the downstream data analysis, where we will be analyzing the temporal trends. Prithvi, can you put the SOP ones? Yes, this is the downstream analysis that we were referring. So here we will be studying the abundance. We will be already having the Excel format. From the Excel format, we need to calculate according to the year wise, according to the location wise, according to the host wise. Those kinds of information we need to retrieve from the Excel sheet. So that can be represented in a different manner. So that we will be explaining it in the next session. Is that fine, Professor Martin and Professor J?
Yeah.
I think it has been a very clear explanation. Thanks for, first of all, for drafting the SOP, and also for the extensive and very clear explanation. What I said, I've made a recording of this, and I will see to it that the recording is being shared with all the people. The same applies to the SOP. I was just wondering also, has it worked out that people have expressed their preferences for the pathogens?
From the Excel sheet, we can see that some of the ambassadors has already expressed their first and the second preference. A few of them are yet to provide their answers. We are expecting their answers soon.
Okay, should I, so
Yeah.
when I'm sending this email, I should also remind them to express that.
In my opinion, you can also send a separate mail, because there's only three has to need to respond. One is Jao, Dr. Jao from Australia, then Stephen and Ansari. So only these three has to
Okay.
express. So I think
Okay,
better to write them directly, so we'll get one answer.
Yeah, I agree. That's perfect.
Yeah.
Yeah. Yeah. Great work, great start. And this must have been a lot of work to
And
prepare this.
one more thing, we should also look for the timeline for this work.
Yeah.
So what is the realistic timeline to get the Excel sheets from all the groups? I mean, any ambassadors can come up with the suggestions. How many days? We have weeks.
Hours.
I know this is a pretty good question, but difficult answer.
Yeah.
I mean, in my opinion, we can also go for 3 weeks maximum. What about others?
Please.
Hello. Are we to stop at the Excel sheet level? Or do we need to do other analysis apart from getting the antibiotic resistance genes?
Maybe can you repeat your question, please? Because
okay, what I'm saying is that the work we are doing for this first phase, is it just to stop at the Excel sheet level where we just show the antibiotic resistance genes?
no, Krishna, you may be the person
Yes,
to answer.
at the first stage, we are executing the tools to get those determinants details, get it done in Excel sheet. And the second step will be the data analysis. In the sense, as I mentioned, if you look at Prithvi, can you put that SOP once again? So we have a raw data, the informations, whether it's a plus or minus, whether it is to what extent it is present. So we need to represent it, isn't it? So that we will be doing it in the next step. Yes, these kind of diagrammatic representation, Dorka. So this we will take up under the downstream data analysis part. Whether to represent the data as a histogram, whether to represent the data in a pie chart, whether to put it in a tabular format, either a country wise, a geographic wise, or source wise. Source in the sense I'm talking about the human clinical cases, animal cases, or environmental.
We call as hosted sources.
Yes. So with this data, we can provide multiple dimensions to the data that we have. So right now we don't have these information in our hand. We have only the genomic informations. From the genomic information, we are moving into the genetic information, the genetic which contributes to the resistance. Once that is done, so it is our responsibility to represent the data in a more presentable manner and discuss the result for that. What we will do is, there are 2 papers that are already appeared. One is on Serratia, another is on Salmonella. Those 2 papers I am going to share with all our ambassadors. And one paper, it is from China, and another paper from United States. It's not a global study. It's a country-specific work that they have done. So with the limited data sets that they have.
Yeah.
So ours will be much more comprehensive, a larger data set with a global perspective.
All right. That's great. Thank you.
Thank you.
So back to the original question. What's the realistic timeline?
For me, 3 weeks.
Okay. What about others? Maskur?
Jay, 3 weeks time will be fine. when we decide the pathogen, when we assign the pathogen, maybe we need 3 weeks.
Okay, from the time of, you are right, from the time of assignment, from that point, 3 weeks. That's a good one. That's a good, correct one. What about others?
If no one disagrees.
Okay, so silence is taken as an agreement.
Can you ask all our ambassador to keep on
No.
their video on? No, keep their video on.
Let's make it
Yes.
1 month, please.
And stop sharing this slide because I see that 2 times your things. Who is sharing?
Prithvi, stop sharing.
Prithvi. Yeah, now.
Sorry.
So all of you can open your video so that we can see that physically, yes. Yeah, Saranya, PK,
Yes, sir.
N-
Internet
M-
is low, sir.
Oh, internet is slow. Okay.
Yes.
Okay, maybe the internet is slow, that is okay. Okay, so no worries. We'll take this as a granted that everyone agrees for 3 weeks from the time we assign or we decide on the pathogen, which group will work. Okay, now I have one more question. In the beginning itself, we decided, okay, 2 groups will work or 2 colleagues will be working on some one pathogen. How this work will be splitted? You got my question? 2 are
Yes.
different geographical location. Different geographical location. That's how we are going to tackle this.
Okay. Whether I need to answer or can we expect from our ambassador itself?
Both are good. Maybe Krishna, I can go ahead.
I haven't thought that particular part, how to do that. Maybe what we can do is maybe if you have a 20,000 genomes, the 20,000 genomes can be divided into 2. And the one part will be done by myself, and another part I will be giving it to Prithvi, something like that. So
Yeah, that's a good idea. But how you are sure that it's exactly split into half? Because you're downloading at different points, different person, different places.
No, exactly not possible. Maybe it will be something like here and there. But when we do the analysis, we will be having a number for that, no? So
Yes,
easy
of
to
course.
match it in the
No.
Excel sheet.
Of course, whenever you download, sometimes depending upon the geographical setting, the
No.
sequence will also change.
I'm telling is download the entire genome data sets,
Okay.
and the entire genome data sets divide into 2.
Yes.
So
Again,
there is
the
a folder
moment
split.
you, yeah,
There
I
is a folder
understand.
split.
Yeah, the moment we download, then it becomes at one location. Then we have to split them
Yes,
into 2.
again we have to split.
Sir, can I give one suggestion?
Yes, go
Yes,
ahead.
please.
Okay, so sir, in Galaxy, I'll just share my tab once.
Hmm.
So here in Galaxy, we have an option to split the pages. So if you create a new history, a new page will be open for analysis. So in 2 different, if you can see here, there are multiple history. So you can run simultaneously in 2 different histories. So what they can do is the person who is having a good internet source among the 2, they can download the entire data set and they can divide it and they can upload in 2 different places. So this they can name for one person, they can name one with their name and another history with another name. Then because the account credentials will be shared to both, they can access from their places and they can simultaneously run the analysis. Because once the data is uploaded, then it's only the job is to run the analysis. So I think that could be the possible
Yeah,
solution.
that was a wonderful suggestion with you. My question is, when one person can download and upload it, why should the other person, why should the data be split into 2 and then 2 different analysis and then finally you need to merge them again?
Because the work with the Excel will be huge to handle the data in
Yeah,
the Excel.
again, if you have, when you have an Excel, 2 different Excel for say Salmonella, 2 different Excel sheets, then again merging these 2 different Excel will be a complicated process, right? Yes. Yes, go ahead.
Yeah. Other suggestions from my side is we are working on a temporal data. So we can also divide the data sets in from 2000 to 2020. Myself will be doing, and all the genome sets prior to that, the Prithvi will be doing it.
No, that's all okay. But when one person is able to upload and do the analysis, why should the other person has to be split? And then again, you will have two different Excel sheets, which you need to combine again.
Yeah, that is there. That part is there.
So what I suggest is that let one person out of the group take over the whole stuff, and the second person let it check the Excel sheet and help them to derive the data in the part 2.
Yes. Yes. That is.
What do you think?
Yes.
Others, what do you think? So it's a pair of two persons will be doing, but one person will upload in the ring and get down the Excel sheets. From that point, the second person can do, and then they can collaborate.
Collaboration. That's also fine. Yes. That's a good idea.
Yeah.
One person will be executing the tool, another person will be collating the Excel sheet and doing the analysis part.
Analysis, because instead of getting 2 different files or 3 different files,
yes,
we can do
that. Yes, perfect.
That one person can take it, and the next part will be the other person. Later, we can also do some other analysis, some other changes or writing part. Everything will come into play.
Yes, that's perfect.
Okay. What about others' opinion? Martin?
No, I'm fine with it. Yeah.
Okay.
Yeah. Makes sense.
So, Maskur, do you have anything?
Jane, it's all right. I agree with your opinion that okay, one should work everything like downloading and then write from the Excel, then the other one will work.
Okay, good. Okay. Okay. So, any other things we need to clarify? So, we agreed for 3 weeks from the time of assignment, and then any other things to be clarified or any other things to be questioned. Of course, from this point onwards, we will have every week, say, half an hour meeting to clarify the questions and doubts. If everyone agrees.
Yes, we can also create a WhatsApp group.
Yes,
and
one WhatsApp group.
Yes, common issues and common things that we can share.
Yeah, what we can also propose is that having a WhatsApp group. What Martin?
Yeah, makes sense. I'm
and
not
then we
happy
can
to set up a WhatsApp group.
And WhatsApp group is only for sharing any information pertaining only to the escape and download. You understand what I mean? Because there's a tendency to propagate something, but we all should not do that. Only one purpose, the exchange of ideas regarding only the project. Okay.
Okay.
So who will collect this phone number and who will create it? Again, the practical difficulties.
I can create a WhatsApp group. It's
wonderful, wonderful, wonderful. So everyone sent their email, phone numbers, mobile numbers to Martin.
Yeah.
And then WhatsApp number, please. Don't send any landline number, and that will not work.
Yeah.
So can we use this chat message, or shall we email our WhatsApp number to Martin?
I think we can email personally. I mean,
yes, that's
that
better.
would be really great.
Yes. Otherwise, it's very difficult to search it from the chat box and copy it to the system.
Or we can also create one Excel sheet and everyone can add it there.
Yeah, that's
Martin
better. That's better.
One click.
Yes. Yes.
Okay.
What we have done is we have already created an Excel sheet to express their organism. There itself, we can make one more column and
one more column, phone numbers. That's all.
Phone numbers. Yes. Yes. I will do it right now.
Yes. That's a great idea.
Yes, I have done already.
Cool.
Just need to enter it.
Okay. Yeah, I see that you have added already. And
we need to add our name.
Yeah, that's what I'm doing. Please, with the country code, please.
Yes.
Yeah, I have already added my number also. Yeah. Okay. So please add with the country code so that everyone knows that.
Yes.
Yeah, I see Mark also started doing that. Ah, this is quite fast. So we got the best team in the world. So once we are connected to WhatsApp, I think things will be much better. The questions can be asked, and we can also fix the meeting and everything through that.
Yeah. Okay. I have to apologize. I do have another meeting right now.
Yes, of course, you're right. We have to.
Yeah.
Yeah. Okay. So anything more? Once we create WhatsApp group, we can also have the discussion there.
Yeah. Yeah. Krishna, Peter Welker, and also Ray, Jay, thank you very much for preparing.
So thank you, Krishna. Thank you, Prateek. Thank you, Martin. Thank you, Maskur, and everyone here.
Thank you, Martin. Thank you, Jay. Thank you, all
Yeah.
ambassadors. Yes, thank you all.
Okay.
Thank you, Prithvi.
Have a great day, and then we contact further by WhatsApp.
Yeah.
Yes.
Okay.
Thank
you.
you.
Bye-bye.
Yeah.
Bye-bye.
Bye.